Okay, next topic is the life cycle of Plasmodium vivax. So far, we discussed about Plasmodium vivax is a protozoan parasite. It is a digenetic parasite. Digenetic means it completes its life cycle in two different hosts. That is called a digenetic parasite. For Plasmodium vivax, the primary host is female Anopheles mosquito, where a sexual reproduction takes place. So it is also known as definite host. Human beings or man is considered as the secondary host because here asexual reproduction takes place. So coming to the life history of Plasmodium vivax. So Plasmodium vivax is a digenetic parasite. Digenetic. Okay. So coming to the life history. Life history of Plasmodium vivax. It has the life history in the man, and second one is mosquito. That means it completes its life cycle in two different hosts. One is man and second one is mosquito. Now our discussion starts with the life history in man. So in man, Plasmodium vivax undergo asexual reproduction. Asexual reproduction. This asexual reproduction in the Plasmodium vivax, it is known as schizogony. It is called schizogony. This is the first point, schizogony. This schizogony in man, it takes place in the two different places. One is liver, organ liver, and second one in the blood, RBC. So schizogony is a asexual reproduction. It takes place in the two different organs. One is liver and in the blood, RBC. In the liver, it is known as hepatic schizogony. Hepatic schizogony. In the RBC, it is known as erythrocytic schizogony. Erythrocytic schizogony. In the liver, in the hepatic schizogony, it undergoes two different cycles. One is pre-erythrocytic. Second one, exoerythrocytic schizogony. So, in our body, Plasmodium vivax undergo asexual reproduction, that is known as schizogony. This schizogony takes place in the liver. It is called hepatic schizogony. It is once again two cycles are there. One cycle is called pre-erythrocytic schizogony. Second cycle is called exoerythrocytic schizogony. In the RBC, it is called erythrocytic schizogony. Now we discuss about the first one, liver, hepatic schizogony. In the hepatic schizogony, we have two different phases or cycles. Cycle number one, pre-erythrocytic schizogony. Okay, pre-erythrocytic schizogony means before entry of the into the RBC. That's called pre-erythrocytic schizogony. Second one is known as exoerythrocytic schizogony. exoerythrocytic zygogony. Next, coming to first one, pre-erythrocytic zygogony. Yesterday in the last class we discussed about the structure of sporozoite. The structure of sporozoite means here female Anopheles mosquito. Female Anopheles mosquito with sporozoites means this is the infected female Anopheles mosquito. When it bites a healthy human being, the sporozoites present in the salivary glands. Okay, in the saliva, the sporozoites are present. These sporozoites, they released into the blood. They enter into the blood. Two scientists are important here. One scientist is Granum. Second is Short. So Short and Granum confirmed that these sporozoites exactly after half an hour, means 30 minutes, within the 30 minutes they reach the liver. The hexagonal hepatocytes, this is the suppose liver cells, these are the liver cells with nucleus. In these liver cells, the sickle shaped sporozoites enter inside with nucleus also here. They enter inside the liver cells by using its apical cup. So the apical cup they secretes some enzymes. Cells of the liver, the sporozoites enter inside. So sporozoite enters inside. After entering into the liver cells, this sporozoite first converted into trophozoite. Trophozoite means it increase in size by taking the food material as liver cells. It eats the content present in the liver cells. So trophozoite stage is the feeding stage. Okay. In the feeding stage, it increases its size and becomes the round shape. That round shape is known as zygont. This zygont undergo divisions and produce means undergo zygogony. Zygont undergo zygogony and produces 12,000 cryptozoites. 12,000 cryptozoites. These cryptozoites also known as, other name for this cryptozoites is known as first generation. They are known as first generation merozoites. They are called first generation merozoites. These first generation merozoites, also called cryptozoites, they enters into the another fresh liver cell. So they attack on the other fresh liver cells they enters into the other fresh liver cells and converted into trophozoite first, then becomes the shy giant. Same process, same process, where they enter inside the first time enter inside the liver cells. Whatever the stages are there, same stages they follow in the fresh liver cells also. Okay, in the fresh liver cells, shy giants are created. These shy giants undergo schizogony and produce other type of the cells. Other type of the cells. These cells are known as second generation merozoites. The life cycle of Plasmodium in the man, it has two phases. Completes its life cycle in the man in the two different organs. One is liver and second one is RBC. In the liver, it undergo once again two stages or two cycles. One cycle is known as pre-erythrocytic cycle. So here up to the formation of cryptozoites, it is known as pre-erythrocytic cycle, and then formation of second generation merozoites. This is called exoerythrocytic cycle. Exoerythrocytic cycle. See here, what we are discussing, the life cycle in the human beings in the liver, that is called hepatic schizogony. Hepatic schizogony is once again two types, pre-erythrocytic cycle and exoerythrocytic cycle. In the pre-erythrocytic cycle, the sporozoites enters into the liver cell and produces 12,000 cryptozoites, also known as first generation merozoites. These first generation merozoites enters into the fresh another liver cells, that is called exoerythrocytic cycle, and they can follow the same sequence: formation of trophozoites, increasing in the size, converting into round shaped shy giant. Shy giant undergoes schizogony and produces second generation merozoites. Okay, so here in the liver, what happening here? Pre-erythrocytic cycle and exoerythrocytic cycle both are completed. So in the exoerythrocytic cycle, whatever the second generation merozoites are there, they are of two different shapes. Some are micro, some are macro, means some are small in size and some are large in size. Pre-erythrocytic cycle or pre-erythrocytic schizogony, for completion of this pre-erythrocytic schizogony, it takes 8 days of time, 8 days, approximately 8 days. Okay, and this exoerythrocytic cycle, this cycle completes within the 2 days of time, 2 days. So totally approximately 10 days. Within the 10 days, this pre-erythrocytic cycle and exoerythrocytic cycle, these two cycles completes. And now what happened here? This is second generation merozoites. Second generation merozoites also known as, these are also known as metacryptozoites. I will take second slide. See here, whatever the so here in the exoerythrocytic cycle, whatever these structures are present, these are known as second generation merozoites. These are also known as metacryptozoites. Okay, these metacryptozoites are, once again, there are two types. One is small in size, one is large in size. One is large in size, one is small in size. Large are known as macro. metacryptozoids. They are known as micro metacryptozoids. Okay, these small micro metacryptozoids, they enter into the bloodstream. They enters into the bloodstream. In the blood, this is the blood vessel. In the blood, they enters into the RBC. They enters into the RBC. Okay, so that is the micro metacryptozoid enters into the RBC and starts the erythrocytic cycle, the second one. In the human beings, Plasmodium vivax can take cytogony in two parts. One is hepatic cytogony, second one is called erythrocytic cytogony. So here erythrocytic cytogony is present in the blood, whereas the hepatic cytogony is of two types or two cycles. One is pre-erythrocytic cycle or cytogony, second one is exoerythrocytic cytogony or cycle. So up to here, whatever the cryptozoids, also known as first generation merozoites, they are attacking the fresh liver cell. Within the liver cell also, they convert into trophozoite, shygiant, and the shygiant undergoes cytogony and produces second generation merozoites, also known as metacryptozoids. There are two types in the size. One is large and small. The small are called micro metacryptozoids. They enters into the blood in the RBC and they start the second one, that is called erythrocytic cytogony. And what about this large one? These large macro metacryptozoids, once again they are attack on the fresh liver cell. Once again they start the trophozoid stage, shygiant and cytogony, and produces the third generation merozoites. So like that, in the liver the cycle continues one side, and second side in the RBC the erythrocytic cytogony also starts here. So in this erythrocytic cycle also, these micro metacryptozoids, they starts this erythrocytic cytogony. Here what they do, so these micro metacryptozoids also eats the hemoglobin present here. Hemoglobin. In the hemoglobin, they eat globin. They eat globin, and whatever the heme is present, that heme is converted into hemozoin granules. Okay, means here this micro metacryptozoids, then they converted into another stage that is called signet ring stage. This name is called signet ring stage. Then they converted into amoeboid stage. This is called amoeboid stage. Amoeboid stage increase in size, and then increasing the amoeboid stage, the RBC also increases in the size of the RBC, and that increasing of the size of the RBC is known as hypertrophy. Hypertrophy is the effect of parasite in the RBC. So what happening here, increasing the size of the amoeboid shape in the RBC, the size of the RBC also enormously increases, and this is known as hypertrophy. After hypertrophy, the amoeboid stage is burst and releases here they releases the hemojoin inside, and along with the hemojoin they also contain small dots are there. These dots are known as Schaffner dots. Schaffner dots. These dots are believed to be their antigens, malarial or Plasmodium antigens. Okay, hypertrophy is the increasing of the size of the RBC. Hemojoin is the chemical. Hemojoin is the chemical that chemical causes or produces the malarial symptoms. So these symptoms, when the symptoms will occur whenever the amoeboid stage burst out, and then the RBC also burst out, and all this content enters into the bloodstream. So whenever the hemojoin granules enters into the bloodstream, then the person shows the malarial symptoms like cold stage, hot stage, and sweating stage. In the cold stage, the shivering will occur. After immediately there is a hot stage. Okay, hot stage, they can sweat more. So like this is a different stages are there here, chill and also hot. That means cold stage, hot stage, and hot stage is followed by this is a sweating stage. So all these will occur and then stops. Means whenever the hemogen enters into the bloodstream, then they produce the malarial symptoms. And this is Schaffner dots. Schaffner dots are believed to be their antigens. They are antigens produced from the malarial parasite that is called Plasmodium vivax. Okay, and after this cycle is continuous. From the to the 2 or 3 generations, and in some of the stages what happened here. So whatever the small amoeboid stage in the mitosis, they produce 12 to 24 cells they will produce. So here in the erythrocytic cycle, this amoeboid stage then produces 12 to 24 erythrocytic merozoites. Okay, these erythrocytic merozoites, they attack on the fresh RBC once again, and they start the same cycle, and exactly at some time 24 to 48 hours they releases their hemojoin in the blood. So like that this cycle is continuing, and at one stage what happened here, the 12 to 24 erythrocytic merozoites are stops to deliver or comes outside, and then this RBC prepares the male and female gametes. So here male and female gametes are created. Male gametes are small in size and female gametes are large in size. So female gametes are large in size and male gametes are small in size. Like this, these two gametes are produced in the blood and waits for the bite of the female Anopheles mosquito. So up to now this one. So what we discussed here, in the liver hepatic schizogony is present. In the blood there is a erythrocytic schizogony. Okay, and these two collectively produces at last in the blood they produces male and female gametocytes, and these gametocytes never fuse and form the zygote in the human beings, and they enters into the female Anopheles mosquito gut or stomach, then only they fuse and forms a zygote. That is why up to now this completes the life cycle of Plasmodium vivax in the man.